So welcome to the end of May. It's already almost the end of the term. How did that happen? It's kind of frightening. So we'll have 2 lectures this week, 2 new lectures next week, and then a review next Friday. And then the final is at 8 on Tuesday the next week. So that's the normal exam time for this class. So as usual, non-cumulative, 50 multiple choice questions, et cetera, et cetera. 2 hours should be more than enough time for you to be able to finish that. As I promised, we have a couple of clicker questions from the last couple of lectures that we'll start out with, assuming that I remember to turn on the clicker software, which I didn't. So it gives you a second to get your clickers out. There we go. Okay, so the first one is what seems to regulate maintenance of latency in HSV1? Removal of the tegument, transcription of latency genes, transcription of VP16, translation of ICP0, degradation of ICP4. Huh? What? That was like forever ago. Again, feel free to discuss this. Ten, 5. Yes, it is the transcription of the latency genes. Removal of the tegument happens when? When you have fusion with the endosomal membrane, usually. What's the tegument? Right, it's the stuff between the capsid and the outer membrane. And what lives there, among other things? VP16. What's VP16? Transcriptional activator. ICP0 and ICP4 are also transcriptional regulators, but all have to do with the lytic cycle of herpes replication. So B it is. Feeling happier? No, yes, maybe. Do another one on herpes. Let's hide these quickly. Oh, no. Okay, you got some extra time here. Late genome replication in HSV1 is most similar to genome replication in which of the following viruses? Adenovirus, SV40, Lambda, MS2, or AAV? That's adeno-associated virus. Stedman's favorite kinds of questions. Where is he? Where'd he go? He's out riding his bike, getting his points. Ten. 1, 0. This is interesting that very similar results. Nobody liked MS2 because why? It's an RNA virus. So very little to do with HSV1. AV, turns out AV replication is actually really similar to poxvirus replication that we'll get back to a little bit later on today. So that leaves with adenovirus, SV40, and lambda. So what's the key about the late genome replication in HSV? And how does maybe another way of looking at it, how does the genome get packaged? Yeah, so it's rolling circle replication gives concatamers, and those concatamers get packaged. That's like what? Lambda, yes. So adenovirus, that's with the protein primed, and so it's got this really interesting panhandle structure because the inverted terminal repeats at the ends. And SV40 is boring because it's just like the way that all of us replicate. Bidirectional replication around a circle. Of course, our genomes aren't actually circular. Okay, so deep breath. Now think about the single-stranded DNA viruses. Okay. The primer for replication of AAV2 is RNA made by a viral RNA polymerase, RNA made by a cellular RNA polymerase, a protein, DNA made by a viral DNA polymerase, DNA made by a cellular DNA polymerase. Ten, 5, 'A little bit more consensus here. So everyone seems to think it's DNA, not protein, which is good. It is a DNA primed. But are there viral DNA polymerases in AAV? The genome's too darn small for their own polymerases. So it's all a viral DNA, which primes its own synthesis, but that's all made by a cellular DNA polymerase. Yes, no, happy nods, shakings of heads, case may be. Okay, so it's a cellular DNA polymerase that does that. Okay, last one of the old clicker questions. The RDHV virus genome that pre-Dr. Kaysen works on probably has a blank package genome, linear DS, circular DS, linear SS, circular SS, none of the above. Oh, maybe I should start, sorry. I'm not used to having all these clicker questions at the beginning either. Getting closer to a consensus. 10, 5, okay, much closer to a consensus here. Yes, it is a circular single-stranded DNA, or at least we think it's a circular single-stranded DNA. We don't have the virus yet, which is really embarrassing, actually. But Certainly, all the indications are that this particular virus packages a genome very similar to circoviruses, which are these single-stranded DNA viruses that infect animals, and not surprisingly that they're called circoviruses. They have circular genomes. It has to go through a double-stranded DNA intermediate, which is one of the things which is really interesting about all of these single-stranded DNA viruses. You have to make a double-stranded DNA. But that's always done after you have release of that genome inside the cell. Yeah?
Can you remind us why they found that little bit of single-stranded RNA in the genome?
So the idea here is with the RNA, which is the quote-unquote RNA and the RNA-DNA hybrid virus, that was historical. So the gene which codes for the capsid protein in this RDHV, or we're now calling them the cruciviruses, that gene encodes a protein that's most similar to capsid protein genes from RNA viruses. And that's where that RNA comes from. So presumably, and this is all hand-waving, is there was at some point a RNA-DNA recombination event where that gene got incorporated into the single-stranded DNA virus genome. But that step, and that's literally what George is working on, is to try and figure out how that works. Yeah?
Oh, could it have been like a virus that attacks other viruses?
So could it be a virus which attacks other viruses? So basically a virus just sort of parasitizing or satellite virus on another virus? In theory, yes. I would say in practice, all of the examples that we've seen of one virus parasitizing on another virus, one is really small, like AAV, and the other one's really relatively big. And when we talk about the Mimi viruses and the Sputnik viruses next week, the virophage, one really tiny, one ginormous. And these, the genome sizes of the presumed ancestors of this crucivirus are both really pretty small. So it's probably a simultaneous infection in one cell that happened between an RNA virus and a DNA virus, and then somehow that DNA virus, the single-stranded DNA virus, stole the gene and incorporated it into its own genome. Very, very bizarre. But that's why we study it, because it's so bizarre and cool. So yes, so the answer is D for this. And there will be some pox virus questions too, but we'll have those a little bit later on. Okay, so pox viruses. These are the big picture. It used to be these were always the biggest viruses that we would ever talk about. Now, of course, people have found the Mimi viruses and some of these giant viruses of amoeba. But basically, this is sort of the largest of the DNA viruses which infect organisms that we care about. So these are quote-unquote linear double-stranded DNA viruses, and we'll see why I have that in quotes a little bit later on here. Sort of following this progression, they're larger and more complicated, certainly in terms of the number of genes that they encode than the herpes viruses. And one of the really fascinating things about these viruses, and also some of the giant viruses of amoeba that we'll get to a little bit later on and next week, these are practically autonomous viruses. And when I say that, they have almost all the machinery in the virion that they need to replicate. So this is a double-stranded DNA virus that replicates in the cytoplasm. It doesn't go to the nucleus at all, which is really crazy. And it gives you a pretty good idea of all of those viral-specific proteins that have to be part of the virion in order to be able to replicate its DNA, to do all of its transcription, to make messenger RNAs, et cetera. What it doesn't have, and no viruses have so far, is what? So it makes RNA, makes messenger RNA, replicates its genome, doesn't have its own ribosome. So no viruses with ribosomes yet. So this is about as close as it starts to get to really be sort of a cell by itself. Yeah?
Are these, do they tend to be the oldest that we've found too? Do they seem to be deepest in the phylogenetic trees?
Okay, so the question is about how deep these are in phylogenetic trees. And we'll probably talk more about that when we talk about the Mimi viruses a little bit later on. There's this whole theory called the fourth domain of life, that some of these giant viruses, it may have been a cellular domain and they've descended from that. Garbage. No, there's pretty good evidence that that's not the case. And in fact, what seems to have happened with a lot of these giant viruses is they've actually accumulated genes from other organisms, much more so than the reverse. And then, of course, why people care so much about poxviruses was an incredibly nasty human disease, killed way more people than all wars combined, and we've completely gotten rid of it, which is absolutely amazing. And because of the reason for that is really all about vaccines, and we'll spend a while talking about that today as well. Also, poxviruses turned out to be a really wonderful model for studying messenger RNAs because, again, they can make messenger RNAs just basically by themselves, and so you don't have to worry about all that ugly nucleus stuff, all the cellular business as well. So a couple of other quick key concepts here: vaccination, 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 vaccination. Probably can fill the whole slide with this. We'll spend a little while talking a little bit more about vaccination and how wonderful this is. In terms of the genome, I mentioned it's a quote-unquote linear double-stranded DNA genome, but you could also think of it as a single-stranded circular genome that's almost completely complementary to itself because it's in fact covalently closed. Each of the ends, it's one continuous DNA molecule that goes all the way around. And so that is these hairpin end structures. And these hairpin end structures should sound really familiar because that's what George talked about when he talked about AAV as well. It's these self-complementary ends, which probably should give you a good idea on how replication gets primed. Interestingly enough, there's zero splicing that happens in all of these messenger RNAs. And the reasoning for that is probably because, again, this is all replicating in the cytoplasm, not in the nucleus, so it doesn't have all of the splicing machinery that otherwise would be in the nucleus. And so all of the messenger RNAs that are made from poxvirus genomes are made without being spliced. And again, this is a really nice reason why it is a nice model system to study looking at messenger RNAs, because you don't have to worry about all the extra splicing, alternative splicing, and so on and so forth. So, one last thing is these so-called mature viruses and enveloped viruses. This is kind of a funky concept, so I wanted to spend a little bit more time talking about it, and we'll go over it again later. It turns out that these viruses can have either one membrane on the outside or two membranes on the outside. Each of these are infectious, and those membranes, and particularly the envelope glycoproteins that are in those membranes, then can go to different targets. And it turns out this one with an extra membrane on the outside is important for cell-to-cell transmission inside an infected organism, and this one is much more important for infecting new organisms, so transmission from one organism to another. And when I say organisms with at least smallpox, there's one and only one host that we know of for smallpox, which is humans, which is one of the reasons we're actually able to get rid of it with vaccines. You have a question? Yeah. How does it uncoat both layers? That's a great question. And it turns out that one of the layers is really pretty unstable, and that's this one right here. And so once it actually gets into the endosome, and this is the endosome process, that is burst, and then you have normal fusion that happens with this membrane. Yeah?
So the inner membrane destabilizes and disintegrates before the outer membrane does?
No, it's the outer membrane that destabilizes, and then the inner membrane is where you have your membrane fusion, sort of classic membrane fusion. But the main thing really seems to be it's these receptors on the outside of the virion, which, again, are really important for going from cell to cell inside one organism. But when you're inside the organism, you don't have to be as tough. You can actually get away, again, to totally over-anthropomorphize here with an unstable membrane. But then when you're transferring from cell to cell, and it turns out these are infamously stable, and that's a different, then those are really quite stable. Yeah?
Could it just be like a byproduct of it being unstable that some of them don't have it and some of them do?
It doesn't seem to be the process, and we'll talk a little bit about that right towards the end of the lecture today, assuming we get there, in terms of how these are actually being formed in terms of the budding process. It's a really fascinating process, thinking about how do you get multiple membranes around a virion is really quite interesting. Okay, so again, as usual, talk about origins, disease, of course, in this case, a little bit about the genome's replication, transcription, and release. But the main thing here really has to do with the disease. This is a viral disease that's been known for just about as long as written history, certainly more than 3,000 years ago. And this was the smallpox versus large pox, which was syphilis. Probably why most of us have white skin here rather than much darker skin, because of all the conquistadors who came over and brought these nasty diseases with them to Central and South America. Killed off most of the armies, et cetera, that were here that probably would have taken things out. Very high fatality rates, particularly among kids, and really, very, very nasty. And they have a quote in the textbook about, no, you really couldn't count a kid as yours until it actually had smallpox and gotten over it. So really amazingly nasty disease. And again, around 25% fatality rate in adults. So just crazily high. What's the reservoir for poxviruses? For the variola virus, which is the smallpox virus, humans are the only ones who are known to be infected by smallpox. And this is another reason why we were able to get rid of it and why we're getting close to getting rid of polio, because it seems to be the only organism that these viruses actually replicate in. There are a number of other pox viruses. Vaccinia is the vaccine strain. As we'll see in just a second, it's been around for literally hundreds of years, and where it originally came from, no one's really quite sure. But what's nice and what allows us to actually work with it is it grows in a number of different organisms. So the variola, again, is extremely specific to humans, but some of these other ones are considerably less so. One of my favorite names of any virus is this one. It's the ORF virus. I have no idea where the ORF came from. I think it's an ovine for sheep. But yeah, a number of these different ones. Don't believe necessarily the names. This is, again, historical in many cases. You'll notice that cowpox here actually probably is hanging out most of the time in rodents. So just relatively infrequently do you have cowpox. Yeah?
This could be completely off track, and so just ignore it. But that name, molluscum contagiosum, the only host is humans. I mean, in biology, mollusks are sea creatures.
Yeah. No, it has to do with the form of the lesion, which actually looks kind of like a little bit of a shellfish thing. So that's the only reasoning for that. Doesn't seem to cause much in the way of disease, just little blobs. So, in fact, my daughter was diagnosed with this at one point.
Really?
Yeah, cleared it pretty easily. Didn't seem to be a big problem. So, got some really nasty diseases and really not much of one at all. So, why don't we have to worry about variola anymore? This has to do with vaccination, but well before vaccination. And this is sort of the very dawns of immunology, when people really didn't have a clue what they were doing. But there were some very smart doctors at that point and basically noticed that if you've been infected once, you didn't ever get infected again. This is also why, if you only count your kid as your kid once they've already gotten smallpox, is then they're completely immune afterwards. And what people figured out is that, and this is mostly in India and in China, that you could take small amounts of some infected lesion, and you remember those little pox that we had before, that ugly picture. There'll be some more ugly pictures later, don't worry. And you can take small amounts of those scabs and use those to infect someone, but just on sort of a surface infection, or in some cases, in fact, intranasal infection. If it's a small dose, what seemed to happen is that in most cases, way better than the 25% death rate or the 40% death rate that you had in kids, about 1% would actually die. Now, any kind of treatment now where you have a 1% death rate, probably not a terribly good thing, but be that as it may, it's certainly way better than the 40% or the 25%. So that was the real kind of breakthrough, and that was known for probably quite a long time, certainly tens, if not as close to 100 years, before Edward Jenner on the 14th of May, so almost the birthday, vaccine day, a week or so ago. Edward Jenner noticed that milkmaids didn't get cowpox. And this is the milkmaids was always the beautiful skin, et cetera, because they hadn't gotten smallpox before and hadn't got all the lesions that were left over from that. Why? Well, he thought might be because that they had a pox virus infection, which then gave them immunity. So he, in fact, tested, poor kid, which I should put up on here, with the vaccination with cowpox, and then did variolation. And then so the real question here is, is this a completely ethically questionable kind of study to do? The kid was going to get variolated anyway. So this was sort of a case of, okay, we can try this before the variolation process. And variolation was, again, it's a 1% chance of dying, but still way better than the 40% if you just ended up getting infected. So, of course, it worked. And since then, we've had vaccines. So 1790, really quite a long time ago. And apparently, for a long time, what they had was, so to do this, you had to have a supply of the vaccine, which is the cowpox. They would actually infect people with cowpox and put them on ships to go somewhere else, and then get those cowpox lesions again and use that for vaccination wherever they ended up at. And vacca, of course, is cow. That's where the whole idea of vaccination came from. Of course, the cowpox, now we've sequenced the cowpox virus, has nothing to do with Vaccinia, or very little to do with Vaccinia. So where exactly Vaccinia came from, or whether it showed up in some of these vaccine stocks, some guy was being sent across the Atlantic to bring the inoculum to the US. We're really not quite sure where all of that came from. So through lots of this vaccination process, which worked extremely well, smallpox has almost completely been eliminated. And in 1978, the WHO made this big declaration: we're done with smallpox, the end of smallpox is over. And what they had planned on doing was having a group at the CDC, Center for Disease Control in Atlanta, and the equivalent in the then Soviet Union, have their vials of smallpox, put them in the autoclave at the same time, and everybody push go, and they would all be gone. That was actually decided that they didn't want to do that, because they thought, hey, well, maybe there's some other smallpox somewhere else, and studying these viruses might actually be a useful thing to do. And it turns out that one of these guys standing here was one of the people who made that decision. It wasn't this guy. And this is one of my colleagues, actually, who has been very involved in thinking about virus structures, so conservation of virus structures. This fellow here, his name is completely escaping me, Antonio. He's a Spanish virologist, works on monkeypox. But he's actually But it was him. No, he's actually on their commission. So every year they get together, the WHO, and decide, okay, do we want to hang on to our variola stocks, or do we want to get rid of them? So I had about a 3-hour car ride back to Madrid from this archaeological site, which is really fascinating and a whole totally different story, talking about why to hang on to the variola or not, as the case may be. So there are, as far as we know, 2 known stocks of variola, one at the CDC, one in, forget exactly where it is in Russia, that we know of. And then, I don't know how many of you heard about, apparently there was a freezer in the NIH that also had some smallpox samples in it that they found 2 years ago. So one of the things about these, that particular, the single membrane form, is incredibly stable. So, you can stick it in a vial, ends up in the back of somebody's lab, and don't go through my lab. In fact, when I moved into my lab here, sorry, getting too many asides here, but when I moved into my lab here at PSU, I inherited it from a different investigator. In fact, the fellow who Ryan Estep worked with. And I opened up the freezer, and there was a box in it that said HIV, and I put that box in the autoclave. I didn't bother opening that particular one. So one thing, again, that I did want to mention is this whole vaccine business. Everyone says, anti-vax is a recent thing. So this is, in fact, in 1812. This was, it was the 1790s that Jenner came up with this. This is supposed to be Jenner himself here, vaccinating people. And it's maybe a little hard to tell here, but what happens? What are the lesions that are coming out of the arms? These are all cows. So, so this is the horrid, and here, the cowpox, or the wonderful effects of the new inoculation. Publications of the Anti-Vaccine Society. So anti-vax has been happening for long periods of time. This is the more modern approach, which, oh no, this wasn't photoshopped at all, I'm sure, never. One of the big arguments that people have regarding vaccines is there are too many vaccines at the same time. Any of you who have kids know what they put in their mouths constantly. They're being exposed to way more antigens than anything that's happening in any of these vaccines. And in fact, what's happening with vaccines is the amount of antigens actually going down in all of the vaccines, and combining them together actually seems to work. You can actually have less of these inoculations that are going on there. One of my favorite images regarding vaccines and the advantages of vaccines is this poster, which is a couple of years old now, that I got from the CDC. And even more striking is that what we're talking about here, pre-vaccine morbidity in just the USA, and now reported cases. So not morbidity anymore, this is just cases. So diphtheria, 21,000, 0. H. influenza, 20,000, a couple of hundred here. Hepatitis, etc. Measles, 530,000. Annual morbidity. Measles in the US now these are just cases of 61. Now it's gone up a little bit. Pertussis, 200,000, etc. But this is hopefully good enough data for most people. Apparently not completely, but this is the example down here of smallpox. Even 30,000 a year in the US, which has now completely gone to 0. So now what I wanted to do is take a little break and show you this smallpox video. The protagonist of this I consider to be really one of the heroes of vaccines and actually really human health at all, Donald Henderson, who unfortunately passed away earlier this year. But let me show you this. In fact, one of the things that makes me the proudest about this particular video is I was included in this same broadcast. Found out later that it was filler that they had to fill in the rest of. But may have a chance to show you that video the very last day. Yeah?
For my epidemiology class, we had to watch a video for smallpox, and they said the hardest part was getting people to admit that they had smallpox. Because a lot of people in India and other countries figured it was like their kid being touched by the gods or something like that. So they'd lie.
Yeah, well, vaccination was a big deal. And one of the main things here, and we'll see this a little bit later on in the video, it's about a 10-minute video, is that getting local vaccinators was absolutely key. And again, getting to that point, as you said, one of the issues was that people thought this was an okay thing. And that's historically, the fact that your kid had smallpox and survived was a really good thing. And so it is completely understandable. So let's take a look at this now.
In the world of virus hunters, this man is a giant. Dr. Donald Henderson accomplished what no man has ever done before. He completely wiped out a virus. And not just any virus, but the deadliest and most feared of them all. Smallpox. The virus has killed 300 million people in the 20th century alone, and it left millions more disfigured and often blind.
It's been said that smallpox has killed more people than all the wars in history combined.
Donald Henderson is still haunted by memories of visiting the victims.
The
Their faces were anxious, there were flies all around. Some of them were crying, some of them you could see were dying. The odor was just overwhelming. It was like rotting flesh. And the British physician I was with put his hands on the railing and he said, I could never do that again. I could never do rounds on these patients. That is the most horrible thing I've ever witnessed in my life.
The battle against smallpox, in fact the outright war on all infectious diseases, started in earnest at the end of World War II. It was an era of science and progress, a time of confidence, some would say of hubris.
There was a tremendous optimism that science was going to protect us and it was going to solve all our problems. Penicillin had just